Awesome. So it's a great pleasure to be here today. And what I'm going to be talking about is basically how energy is actually conserved in Antarctica and how carbon is actually fixed in Antarctica. So I'm going to present this hypothesis that potentially chemosynthetic carbon fixation rather than photosynthetic carbon fixation is the primary way that Antarctic microbes actually generate energy and fix carbon, and in turn that's what's actually sustaining life in this continent. So for various reasons, what we can see is that when we look in Antarctica, there's all sorts of microbial life present despite incredibly huge physicochemical pressures. So when we look at the ice-free soils of Antarctica, for instance, normally we see that there's actually quite a vast diversity of both bacteria and archaea there. For instance, in this study that we recently put on bioRxiv on the Mackay Glacier region, we were able to get over 400 genomes of microbial genomes from 20 different phyla in ice-free soils where there's such minimal water that to sustain photoautotrophs, and there's very little carbon to sustain heterotrophs. So for some reason, nonetheless, life can exist there. So we wanted to figure out why. Likewise, there's plenty of life even in extreme environments such as below the Ross Ice Shelf. So here, an expedition led by the University of Otago was able to get microbial samples at regions 400 and 700 meters below the ice. And again, they found that there was all sorts of life there as well. And this is an environment where obviously there's a huge ice sheet that's sometimes up to 300 meters thick. And yet, nevertheless, although light cannot penetrate, somehow life survives there. And moreover, there's also life in the atmosphere too. So when we look at high and low altitude air, we can see that there's both functionally and phylogenetically diverse life there. And this is sort of circulating across the atmosphere, including in Antarctica. So this is a study led by Steve Pointing, who's been leading this global atmosphere survey. And indeed, Antarctica isn't alone in having plenty of microbial life in its atmosphere as well. So we wondered, how is life able to exist in such extreme environments? And we wondered whether chemosynthesis is the missing link. So microbes, for those who aren't aware, are actually highly metabolically diverse. So some of them photosynthesize like plants do, others eat organic carbon like we do, but many also feed on various inorganic compounds like hydrogen, carbon monoxide, ammonium, and whatnot. But also many can also use unusual organic compounds too, including methane. And so when people think of chemosynthesis, normally they think of environments such as this one. So these sort of geothermal hot springs or alternatively hydrothermal vents, where there's obviously huge amounts of seepage of hydrogen, sulfur, and other things going on. Antarctica, on the other hand, hasn't received any attention really at all for chemosynthesis, but and that's largely because people don't think the compounds there are inorganic compounds there to actually sustain chemosynthesis. But what I hope to convince you today is that this is probably not the case, and actually there's a low but consistent amount of inorganic energy sources such as hydrogen, carbon monoxide, and ammonium that's actually sustaining life in this region. So this is the study on the Ross Ice Shelf, and I won't talk about it in great detail. And this has been led by the University of Otago, so two researchers, Federico Baltar and Sergio Morales. And as Melody sort of hinted, I haven't personally been to Antarctica. What I generally am is a sort of data monkey here who sort of gets cool data sets from other people and then tries to sort of figure out what's going on in terms of metabolism, in terms of microbial biodiversity there. And so what they were able to do is drill a ginormous hole through the Ross Ice Shelf, and then they were able to sample the microbial communities below at depths of 400, 500, and 700 meters below the surface. And what they were able to see is that despite light not penetrating through the ice, obviously, nonetheless, there's all sorts of microorganisms there. So not only is microbial life diverse, but it's also abundant and active in this ecosystem. And so what is possibly sustaining them? So what we did was use whole community genomics, so metagenomics, to basically figure out what the microbes in these environments were doing. And we're able to see that there was a whole diversity of approaches. So some microbes were able to use ammonium, others sulfide, others carbon monoxide, both to generate energy, but also fixed carbon. But when we actually looked at the whole community gene expression, so we used metatranscriptomics, what was really interesting is the ammonia oxidizers shown here, those were the ones that really popped out. So what we're able to see is that these were actually extremely abundant, extremely highly expressed in this environment, despite being in moderately low abundance. So what we hypothesize is that the ammonium is actually quite abundant in this environment, largely because of accumulation and basal sea ice. These ammonia oxidizing archaea are able to use this, and in turn they generate energy and they generate carbon, they fix carbon from this process. So this is really unique in two ways. So first of all, this is a marine ecosystem where inorganic rather than organic energy sources are principally driving things. So rather than photosynthesis being the main source of energy, instead you have inorganic compounds. But also what's really fascinating is that archaea seem to be the base of the food web here. So nitrosocumulus species in particular, whereas in most environments you would have various algae and cyanobacteria as a sort of base of the food web instead. So now I want to switch to our work on terrestrial Antarctica, and in particular this discovery that I made during my PhD that bacteria can basically live on air. So my PhD was basically about figuring out how bacteria that are able to persist under adverse conditions, how these were actually able to basically conserve energy during this time. And so I focused on a species called Mycobacterium smegmatis. So this is a soil organism. Some of you might be able to infer from its name too. It has other niches as well. And what this bacterium is notorious for is basically being able to persist literally for years in the absence of its preferred organic substrates. And we wanted to figure out how to do this. So a key finding from my PhD was actually when bacteria go from this growing state to this persistent state, rather than actually just scaling down the metabolism as you might expect, instead they actually broaden their metabolism. So they actually switched to using compounds that they wouldn't normally use for growth, including, interestingly, hydrogen and carbon monoxide. So what I was able to show was that this bacterium was able to actually use atmospheric hydrogen and atmospheric carbon monoxide as alternative energy sources. So basically that means that if you have a flask of Mycobacterium smegmatis with media in that flask, once this bacterium has stopped growing, it's still actually taking up exogenous energy sources, but rather than taking it up from the media, it's taking it up directly from the air, which is so simple and in some ways so obvious, but at the time it was completely unprecedented. And in many ways, it still blows my mind to this day that this is actually possible. So this process enhances growth, as well as critically it contributes to survival during long-term starvation. So this is just some of the pure culture evidence. So this is focused on carbon monoxide. Carbon monoxide to us is toxic, but for bacteria, they actually find it a very valuable fuel source. So carbon monoxide is an incompletely oxidized form of carbon. So once it's completely oxidized, it's CO2 that can release a lot of energy. This bacterium, Mycobacterium smegmatis, is able to oxidize this carbon monoxide to below this dotted line here, which is the atmospheric concentration of carbon monoxide. And this depends on an enzyme called the carbon monoxide dehydrogenase that when we knocked it out, as you can see in the red here, these bacteria are no longer able to consume carbon monoxide. And so what we're able to show is that they're not simply detoxifying this gas, instead they're actually using it productively for aerobic respiration. So they actually get energy from this process. Moreover, when you knock out the enzyme to use carbon monoxide, these cells don't do very well at all. So they have about a 50% reduction in long-term starvation during once they've run out of their organic carbon sources. We saw very similar things for atmospheric hydrogen as well. So what does all this mean for Antarctica? Well, during my DECRA project, which I did in the School of Biological Sciences when Stephen was heading it, what I really aimed to do was understand the wider implications of this process. So we wanted to look at the environmental, ecological, medical and biotechnological significance. What we now know from primarily unpublished work, with some of it being published, is that most soil and marine bacteria can in fact live on air. So they're able to use these atmospheric trace gases, hydrogen, carbon monoxide, to take over. And moreover, these gases even allow bacteria to survive in the most barren areas where plants and algae can't exist. So that includes volcanic craters, but also includes the likes of these Antarctic soils. So some of you will be familiar with this Nature paper, which was published a few years ago with Belinda Ferrari at UNSW as the lead author. And what Belinda's key question was how do soils from the Westfoot Hills and Windmill Islands region, basically, how did the bacteria in these soils basically take over? And we used a combination of genomics alongside biogeochemistry to kind of figure this out. And so we were able to recover complete genome, well, near complete genomes of several of the most abundant bacteria in this ecosystem. And when we looked at the metabolic capacity of these bacteria, they looked remarkably similar to Mycobacterium stromatus. So we saw this wide capacity to use organic carbon. We saw that most of the bacteria were aerobes, so they used oxygen as the electron acceptor. Interestingly, they also had this hydrogenase to use hydrogen, this carbon monoxide dehydrogenase to use carbon monoxide, but also this unique lineage of rubisco, which enables them to actually fix carbon using these trace gases. So you would have heard Sharon already talk about rubisco in plants, but some chemosynthetic microbes have it too, and that allows them to do this process of growing on hydrogen and carbon monoxide. And indeed, we're able to validate that these bacteria in these soils were able to consume hydrogen and carbon monoxide to very, very low concentrations, so well below the atmospheric levels, and that the rates of this were pretty astonishing given the low biomass of these samples. So they were theoretically sufficient to sustain about 10 to the 7 to 10 to the 8 bacteria per gram of soil. So we believe that this is sustaining not just persistence, but also some slow growth of these bacteria as well. So we've since basically extended these findings to other ecosystems too. And what we now know is that trace gas oxidation yields three things in really short supply in Antarctica. So first of all, energy. So as we all know from the Hindenburg disaster and things like that, hydrogen releases much energy. When it's combusted, microbes are actually able to oxidize hydrogen in a productive way and use that for cellular work. Likewise, carbon monoxide is also a really great energy source. And so what these microbes are always doing is basically continually consuming this atmospheric hydrogen and carbon monoxide, which while in trace amounts is constantly there. The atmosphere is this vast reservoir, and then that allows them to basically maintain energy needs for maintenance, but also for a subset of bacteria, they can also fix carbon using this process. So there are these chemosynthetic prime producers that couple the oxidation of hydrogen and carbon monoxide to the fixation of carbon dioxide into biomass. And the latest work we've done is actually look at water. And here we were able to see that cellular water is produced as a result of hydrogen oxidation, which to anyone who knows about hydrogen-powered cars would be no surprise. So that when you combust hydrogen, basically the end product is water, but the amounts of water that's produced by this process are staggering. So based on the rates of oxidation we saw, we believe that 1.1 million water molecules are produced per cell every single minute by this process in these Antarctic ice-free soils. So not only is this a way that cells are able to stay hydrated, but we think it's a quantitatively significant addition to the hydrological cycle in these environments. And so since then, we've extended studies to other regions as well. So we've been able to look at the Mackay Glacier and Antarctic Peninsula regions. And here we always see all sorts of microbes present, but trace gas oxidizers are always the most abundant community members. But as you can see, there's all sorts of other things there, and this can vastly vary between different soils as well. So there are these trace gas oxidizers, but they're often alongside sulfide or methane or ammonium oxidizers, as well as a small but often prevalent range of photosynthetic microbes as well. But perhaps what was especially surprising by this latest study, which is now available as a preprint in bioRxiv, is that there's also predatory bacteria here, like Bordetella vibrio, as well as some parasites too, like chlamydia. So basically there's two ways to sort of make a living in these Antarctic soils. Either you can be highly self-sufficient and do things like use trace gases to make energy and fix carbon literally out of thin air, or alternatively you can basically depend on others and adopt a more parasitic lifestyle. So going forward with SAEF, we see so many wonderful opportunities here to really advance some of these questions. We're really keen to look more about the drivers of microbial community assembly, what processes control energy, carbon, and nitrogen acquisition, but also get a better understanding into the symbiotic and trophic relationships of microbes, and also sort of taking a leaf out of Sharon's book, also getting a better understanding of how Antarctic microbial composition responds to local and global change as well. But we're really flexible and we're really keen to collaborate across taxa and across objectives, and including if anyone has the need for microbiologists in themes 1 and 3, we'll be incredibly interested in talking to them and potentially developing collaborations there as well. And so my team can bring a range of things to SAEF. We got quite good at biodiversity analysis, largely thanks to inspirations of and collaborations with Melody and Stephen. So we don't just do this descriptive stuff, but we can also get a good understanding of turnover as well. We're increasingly doing meta-omics as well. We do some biogeochemistry, but I think we can really benefit from the expertise of some people in SAEF and actually turning this from sort of book analysis into actually getting a much better cell-specific understanding. And finally, there's some capacity for us to do cultivation too, if anyone has an interest in that side as well. So I think I've covered everything I want to say. Thanks so much for this opportunity, and thanks so much to all of my team, as well as these brilliant collaborators. Thank you.